Good morning, class. Today we are going to talk upon heme catabolism, that is the breaking down process of heme. What are the products which are going to be formed during the breaking down process of the heme, otherwise known as it is a bilirubin formation or bile pigment formation. So by the end of this session, phase 1 student should be able to describe the steps involved in the heme catabolism, otherwise known as bilirubin synthesis. If the question comes as heme catabolism, also the same answer. If the question comes as bilirubin synthesis, also the same answer. Right? And then the next objective would be to know the conjugation of the bilirubin, which is going to take place in the liver, and to discuss about the hepatic circulation and even to know about hyperbilirubinemia. So the end product of heme catabolism are bile pigments, and bile pigments are two in nature. One is biliverdin and the other one is bilirubin. Biliverdin gives a greenish yellow in color and bilirubin is yellowish in color. These two are the breakdown products of heme, and these are the colorful excretory products which are formed from heme catabolism. So senescent RBCs, that is the old RBCs, break down liberating heme and globin. Globin peptides are going to be hydrolyzed and the independent amino acids are channeled into the body amino acid pool for its reutilization. Then whatever the iron is going to be liberated from heme, even that iron is also reutilized in the heme synthesis or the formation of other heme containing proteins. The porphyrin ring is broken down in reticuloendothelial cells which are present in the liver, spleen and again in bone marrow. Okay, so where is that breaking down process of heme is occurring? It is in reticuloendothelial cells which are present in liver, spleen and bone marrow to form into bile pigments. The ultimate bile pigment formed is the bilirubin. So there is an equation given. 6 grams of hemoglobin is going to be broken down per day in every person. Right? So out of that 6 grams of hemoglobin which is going to be broken down is going to give rise you 250 milligrams of bilirubin. So this is the equation. Just like that what we have given 60 milligrams of tryptophan is going to give rise to 1 milligram of niacin. Similarly here 6 grams of hemoglobin when broken down is going to form into 250 milligrams of bilirubin. Out of 300 milligrams of bilirubin formed per day, 80% is obtained from destruction of the RBC, 10% from ineffective erythropoiesis, and 10% from degradation of myoglobin and other heme-containing proteins. Okay, so roughly about 300 milligrams of bilirubin is going to be formed per day. Out of that, 80% is formed from destruction of the RBC, majority, right? 10% from ineffective erythropoiesis and 10% from degradation of myoglobin and other heme-containing proteins. This bilirubin does not hold any function in the body, thus it has to be excreted out from the body. So whatever the products which are going to be formed during the catabolic process, which does not have any function in the body, are ultimately to be excreted out from the body. So in that, one of the molecule included here is bilirubin. So how bilirubin is going to be formed? There are two steps involved in this process of bile pigment formation. First one is synthesis of bilirubin or degradation of heme. Both means the same, right? Catabolism of heme, degradation of heme, synthesis of bilirubin. All means the same. Now, heme is primarily degraded by microsomal enzyme system, that is heme oxygenase, and this enzyme requires molecular oxygen. NADPH is the coenzyme along with the presence of cytochrome C. Okay, and this enzyme is going to be induced by heme. So whenever the availability of heme is there, this enzyme is going to be induced, that means produced to take part its action. This is a monooxygenase enzyme, specially catalyzes the cleavage of alpha methynyl bridges, okay, which is linking the independent pyrrole rings. So this alpha methynyl bridges are quantitatively liberated as carbon monoxide. So what is the molecule which is going to be liberated during the formation of biliverdin is carbon monoxide. The iron liberated is oxidized to +3 form and is taken up by transferrin, which is the transport protein. The linear tetraperol formed is biliverdin, which is green in color. So let us try to see in the equation form. Okay, here it goes. Hemoglobin in the macrophages or reticuloendothelial system or the reticuloendothelial cells broken down to form into heme and globin. Globin gives rise to its independent amino acids, and these independent amino acids are going to be either metabolized or reused. Then the leftover non-protein part is the heme. So here this heme will give out its iron, and iron is reutilized. Then the whatever the molecule is there, that molecule is going to be acted upon by heme oxygenase, liberating iron, and that iron is oxidized immediately to form into +3 with the liberation of carbon monoxide, and NADPH is going to be utilized here for the breaking down of the cyclic ring, liberating NADP, and the linear structure formed is biliverdin, which is green in color. In mammals, biliverdin is further reduced to bilirubin. So biliverdin, again NADPH dependent enzyme, biliverdin reductase, converts biliverdin into bilirubin. A red yellow pigment by NADPH dependent enzyme, biliverdin reductase. Whereas in birds, amphibians and rabbits, they are excreting green biliverdin as such. So there is no second step of this reduction possible in birds, amphibians and rabbits. Only this is happening in mammals. Biliverdin acted upon by NADPH dependent enzyme biliverdin reductase to form into the final product, which is the bilirubin, which is yellowish, reddish yellow pigment. To narrate again, heme acted upon by the enzyme heme oxygenase, liberating iron in +2 form. Immediately that is being oxidized to +3 and taken up by the transferrin, and the breaking down of ring structure happens, forming into linear biliverdin by the enzyme heme oxygenase. Methynyl bridge is quantitatively liberated as carbon monoxide, and this biliverdin in humans, it is going to be acted upon again by NADPH dependent enzyme biliverdin reductase, finally to form into bilirubin. Biliverdin is greenish pigment and bilirubin is yellowish pigment. So to show pictorially ineffective erythropoiesis or myoglobin or cytochromes or the senescent RBCs, right? They enter into RA system, liberating heme. Heme acted upon by the enzyme heme oxygenase, giving rise to biliverdin. Biliverdin acted upon by the enzyme biliverdin reductase, forming into bilirubin. Now, whatever the bilirubin is formed, that is unconjugated bilirubin. And which is, whenever I say unconjugated bilirubin, it is not water soluble. So it is lipid soluble. Now, lipid soluble or water insoluble product has to be converted into water soluble product for its transport across. Now, this bilirubin is taken up into the liver. In the liver, there is one enzyme called UDP glucuronide transferase. Taking up UDP glucuronate converts unconjugated bilirubin to bilirubin monoglucuronide initially by the action of the same enzyme UDP glucuronide transferase. It is bilirubin diglucuronide is going to form, and that product bilirubin diglucuronide is called as conjugated bilirubin. Okay, so where is this conjugation happening? Conjugation is happening in liver. Now, this conjugated bilirubin is secreted into the intestine through bile juice. Right? So, in the bile juice, through bile juice, this conjugated bilirubin is going to be secreted into intestine. So, in the intestine, 80% of that going to be converted into stercobilins, and that stercobilins is going to be excreted out through urine. Right, and 20% is going to be converted into urobilinogen, and this urobilinogen, little bit of the urobilinogen is going to be excreted out through urine. Okay, and little bit is taken up again by into the liver, and that is called as enterohepatic circulation. Okay, so this enterohepatic circulation is more dealt in physiology also. Right, so intestinal flora which is present in the intestine convert conjugated bilirubin into Sterobilinogen, and from there to stercobilinogen to stercobilin. 80% of this is going to be excreted out through feces. Okay, and roughly about 20% through urine in the form of ureobilinogen. Little is being reabsorbed into the circulation to be transported into the liver again. This process is known as enterohepatic circulation. So liver plays a major role in further disposal of the bilirubin. Bilirubin formed in RE cells is insoluble in water and which is lipophilic, and this has been taken up by albumin. Each albumin molecule takes up 2 bilirubin molecules, forming into albumin bilirubin complex, and with that it reaches to sinusoidal surface of the liver. Into the liver, it is through carrier mediated active uptake transport system, it enters into the liver cells. Then conjugation happens in the liver. Inside the liver cell, bilirubin is conjugated with glucuronic acid. In the presence of the enzyme, UDP-glucuronide transferase, initially bilirubin monoglucuronide is formed. Later on, it is the bilirubin diglucuronide is formed, which is the conjugated bilirubin. 80% of it is in the bilirubin diglucuronide form, 20% is in the monoglucuronide form. Certain of the drugs like primaquine, novobiocin, chloramphenicol, androgens, and pregnandiol. These are the drugs which can interfere with this conjugation process, which may lead to the formation of jaundice or the occurrence of jaundice. The same thing again to show pictorially, bilirubin through bilirubin albumin complex reaching into the liver. In the liver acted upon by UDP-glucuronide transferase. The first product formed is bilirubin monoglucuronide, again acted upon by the same enzyme. Bilirubin diglucuronide is formed. In the bile, which is going to be stored in the gallbladder, and depending on the need, it is going to be secreted into the intestine. So water soluble excretion of the bile, water soluble conjugated bilirubin is excreted into the bile by an active process, and again is the concentration gradient which is going to be liberated into or secreted into the intestine. So the same one, because it is the enzyme importance, I have shown once again over here. The same reaction converting unconjugated bilirubin to bilirubin monoglucuronide and bilirubin diglucuronide by the action of the enzyme UDP-glucuronide transferase. So this is the entire process breaking down, ultimately forming into unconjugated bilirubin picked up by albumin, entering into the liver. Conjugation happens through the bile duct, it is secreted into the intestine. In the intestine, it is by the bacterial flora action, it is going to be converted into stercobilinogen, and stercobilinogen is formed into stercobilin and excreted out in feces. Little bit is transported through the portal vein into the liver, that is the enterohepatic circulation. The same one, the conjugated bilirubin reaches the intestine, bacterial flora containing the conjugate enzyme, that is the beta-glucuronidase, liberating urobilinogen, and this urobilinogen converting into stercobilinogen to stercobilin. Right, this is again the pictorial representation of the bilirubin transport, the same one. And then the intestinal flora will convert that finally into stercobilin, and stercobilin is going to be excreted out. One more pictorial representation, what exactly is happening. Unconjugated bilirubin is going to be conjugated in the liver. This picture is more in favor of showing the enterohepatic circulation. Fine. Bacterial enzymes convert the conjugated bilirubin into stercobilinogen, stercobilin to stercobilin, and excreted in the feces. Coming to little bit regarding the normal ranges. Total bilirubin normal range is 0. There is a type mistake over here. Total bilirubin is 0.2 to 1.0 milligrams per deciliter. Unconjugated is 0.2 to 0.8 milligrams per deciliter. Conjugated is 0.0 to 0.2 milligrams per deciliter. If plasma bilirubin increases more than 1 milligram per deciliter, is called as hyperbilirubinemia. Okay, in between the levels of 1 to 2 milligrams or 1 to 3 milligrams is called as latent jaundice. Once it crosses 3 milligrams, it diffuses into the tissues, producing yellowish discoloration of the sclera, conjunctiva, skin, and mucous membrane, resulting in jaundice. Okay, icterus is the Greek term for jaundice. Okay, colloquial term what we use commonly is a jaundice. Is that okay? These normal ranges are very important. Please make a note of that. Total bilirubin is 0.2 to 1.0 milligrams per deciliter. Some textbook even have given total bilirubin as 0.1 to 1.0 milligrams per deciliter. Even that also be considered as a normal. Fine. So this is in nutshell regarding the formation of the bilirubin and the normal ranges of total unconjugated and conjugated bilirubins. Generally, we estimate in the lab that total bilirubin and conjugated bilirubin, unconjugated generally is a calculated parameter what we are going to give the reports to the clinician. I stop here. Thank you very much.